Hello and welcome to my channel Be Yourself. This is Dr. Rajni Sharma and today I am here with the third part of the explanation of answer key of part B CSIR JRF Net Life Sciences. So let's start. The first question or the 34th question from the part B is that which of the following are not transcribed by RNA polymerase 2? So the answer will be here tRNA 5S RNA because this is transcribed by RNA polymerase 3. I know you all of you will be knowing the answer. So let's start the other question. Now RNA editing, a post-translational process, is achieved with the help of guide RNA or g-RNA. Which one of the following statements about the process is true? Before answering this question, I will discuss about the guide RNA or gRNA, actually what it is. And I know after explanation, you yourself will be able to answer the question. So let's start. Actually, guide RNAs or gRNA are the RNAs that guide the insertion or deletion of uridine residues into mitochondrial messenger RNA in kinetoplastid protist is in a process known as RNA editing. It is actually post-transcriptional modification of RNA. As you have seen, I have discussed some of in my videos in my channel during RNA processing that how editing or the capping and polyadenylation is there. Like that, it is also a type of RNA editing, but in case of protist. Kinetoplastids is actually a class of plastids which have only one messenger RNA, messenger RNA, I'm telling, mitochondria, having a single mitochondria. Okay. Suppose that this is your protista and this is having the mitochondria here. This mitochondria or the protist mitochondria have two types of circles known as maxi circles, which are about 20 to 25 numbers are there in the maxi circles, and 10 to 20,000 mini circles are there in the mitochondria. And the maxi circles encode for the gene and some cryptogenes. Cryptogenes are actually unusual types of genes present in protist, and the mini circles especially code for the gRNA or the guide RNA. And both of these are clumped in the DNA and are situated at the base of the flagellum. The as I have told that maxi circles has most of the genes that get transcribed, but all the transcript of the maximum of the transcripts of the maxi circles are not translated into the protein. This is because they need some editing before the translation, and the transcript are only expressed in their protein after the RNA editing. So let's see how this happens. Suppose this is the transcript of the maxi circles or the messenger RNA. When we will hybridize it with the guide RNA, what we will see, you will be able to see that this is the lookout guide RNA. Actually, this is the region which has adenine in their sequence. So I have told that gRNA guide the deletion or addition of the uracil. So what happens? This gets looked out because this strand is lacking here the U. So what to fulfill this? What guide RNA will do? It will first cut out the transcribed messenger RNA from here using some endonuclease. Then after cutting it, what you will see that now this strand and this portion are free to move apart. So they will move apart, and the portion which get blank here will be completed by the addition of U. So this is how guide RNA edit the RNA after transcription, either by deleting the extra uridine or the addition of the uridine. Okay. Now, some more points about this is that once the editing is done, the segment can be re-edited by another guide RNA. And during evolution, such type of edition can be lost, though the loss of the edition is lethal in most of the cases. Actually, re-editing is there because the g-RNA is very small sequence, though I have shown it in the large way just to just for the explanation, but g-RNA is very small strand of the RNA. So for the proper editing or the fulfillment of the need of uridine, there is a requirement of multiple, or you say multiple editing is there. So one guide RNA can't fulfill this, so there will be re-editing after once it get edited. So this process can happen in 2 to 3 times for the completion of whole the process of editing. In case of protozoan Leishmania talonale, about 12 of the 18 mitochondrial genes are edited using this process. One of the example of this gene is CYB. Now, next question. Oh, sorry. The question came back to the question that is about the guide RNA. Now, look at the first option. gRNA dependent RNA editing happens in the kinetoplast DNA. This is true. This happens in the DNA. And next option is gRNA is involved in chemical modification of tRNA. This is not there. Guide RNA involved the modification in messenger RNA, not the tRNA. So the answer will be 2. Sorry. So the answer will be, here it will be 2. And the process involves insertion, deletion of uridines is true, as splicing can be re-edited. As I have told, it can be re-edited because the guide RNA is very much small, and to fulfill the requirement of the correct strand for its translation into protein, it can be re-edited. Move to the next question, that is telomerase, a RNA protein complex which completes the replication. Telomerase during DNA synthesis is a specialized RNA. Actually, it is a RNA dependent DNA polymerase. How? Let's have a look about this. In case of eukaryotes, after replication, we get the RNA primer, because we know that this is the leading strand, this is leading strand, and this is the lagging strand. Okay. So what happened in case of lagging strand? It passes in a Okazaki form, and with each Okazaki fragment there is an RNA primer, and after the replication it gets removed, and it is then fulfilled by, or it is the gap was filled by different enzymes into a complete DNA. But if you will see the last end of the lagging strand, what happens? It has the phosphate group at the 5' end, and there is no hydroxyl group at the end to have act as a primer for the replication in case of eukaryotes, and this terminal is known as telomerase. Okay, have a detailed look about this. This will look something like that, like this, because the telomerase of eukaryotic DNA is rich in GT sequence. Okay, now how this get overcome? How this extra end or it will be replicated? Actually, what happened? I am zooming it out, and this is the telomere. And what happened? There is an enzyme telomerase which has a short RNA sequence which will replicate, or will initiate the replication from this 3' to towards some more distance. Okay, look like this. Now DNA get synthesized for some segments more. Now what happened? Now it has, now we can add here the primer or the RNA primer here. It has been added. Now, as this is a case of eukaryotes, so As you have seen in case of prokaryotes, RNA was removed and the gap was filled by the polymerase 1, which was involved during the nick translation. But in this case of eukaryotes, DNA polymerase alpha will act here and it will synthesize the strand. It's the complementary strand. Now the DNA strand which was not there, or that was a telomere, get fulfilled now. So now the originality of the DNA strand get maintained during each cycles in case of eukaryotes. Now, next question is apical ectodermal ridge induction is essential for tetrapodal lymph development. Which one of the following is not essential for the formation of a functional limb? Actually, what happens? Apical ectodermal formation is somewhat like that. This is the point where the limb formation has to takes place. It is very specific, because you know, if it will not be specific, how you will look like if the limb formation will take place from some other part of your body. What happened? First of all, TBX will induce the synthesis of fibrogen growth factor 10 in the mesenchymal cells, and it will then induce the synthesis of EER, or the apical ectodermal ridge formation, from the distal part of the limb's bud. As this process is very specific, so the position of the limb buds and hence EER is specified by the expression boundaries of Hox gene in the embryonic trunk. Now, move to the question. The options are TBX genes. I have told the function of TBX gene and the WNT genes. What happened? WNT acts as a middleman in the positive feedback loop between the ER and limb mesenchyme. Second option is androsterone. I haven't discussed about this. And third is the apoptotic gene. Actually, apoptotic genes is required where suppose this is the limb formation, and during the formation of digits, or you can say the fingers, the cells in between these get undergo apoptosis to give a digit-like structure. Okay, and the fibroblast growth factor, I have discussed about the FGF10, which was the fibroblast growth factor. So the option in this question which is not essential for the formation of functional limb will be androsterone. Hope you have understood it. Next question is, which one of the following best describes the simplest pathway of water flow from the epidermis to endodermis in a plant root? Before that, let's have a look about the simplest pathway. This is suppose the plant cell, and simplest will have followed the path across the plasmodesmata like this. Sorry, like this. It will follow across the plasmodesmata. This is the plasmodesmata, and likewise there are two other pathways too, like the vacuolar pathway. It also have similar route for the transportation like that of the simplest, only the difference was that it passed across the vacuoles. And third is the apoplast, which passes through the cell wall. So what will be the answer? Water moves through the cell wall? No, it is not moving through the cell wall during the simplest movement. Water travels across the root cortex via the plasmodesmata. So this will be the answer. Next question is, the herbicide dichlorophenyldimethylurea is the inhibitor of. Actually, DCMU, or dichlorophenyldimethylurea, blocks the plastoquinone binding site of pigment system 2, which is normally occupied by QB, and it doesn't have any effect on pigment system 1 or 700. So what will be the answer? Answer will be it will affect or transfer the electron from the P680 to P700. Okay. Next question. This is very simple question, that is, which one of the following compound is not a part of alkaloid class of secondary metabolites? This is obvious, it will be lignin, because lignin is the structural material in the support tissue of vascular plant and some algae. Next question is, which of the following statement is wrong about the megasporocyte? So let's see about what the megasporogenesis is. Actually, in case of angiosperms, there are three ways of development of megaspore, which is the female gametophyte. Monosporic, bisporic, and tetrasporic. What happens in monosporic? Uh, Cell plates form two times in the during the cell division in during mitosis. So there will be the formation of 4 cells, of which 3 cells get degenerated and only one develop into female gametophyte. And in the bisporic, only one cell plate will form, and it will lead to the formation of 2 uninucleated cells, of which one get degraded and one will develop into female gametophyte. But in tetrasporic, there is no formation of cell plate, and the cell will develop into 4 nucleated cell. And after mitosis, these cells will develop into the 8 cell stage female gametophyte. Now, how this happens? It will develop into 8 cell stages, of which the 2 cell get migrate at the center and the 3 toward the poles. And after last, when you will look the structure, it will be the embryo sac will look like something like this. This is the chalazial end, micropylar end. The one which has migrated to the nucleus, which migrated at the center, is known as polar nuclei. Toward the chalazial end, known as antipodes, podals, and the one which has migrated toward the micropylar end has 1 egg cells and 2 synergids. Okay, so now move to the question again. The first option is the megaspore developed within the megasporium of the ovule. Yes, this process occurs within the ovule. Megasporocytes undergo meiosis to produce 4 haploid megaspores. It's also true. And all the 4 megaspores, and after mitosis, it develops into 8 cell stages. Okay, so this is true. All the 4 megaspores undergo several mitotic divisions to form female. Here the option is all the 4 megaspores, but you have seen not all the 4. Rather, in the case of monosporic, of the 4, 3 get degraded and only 1 develop into the female. So the answer will be, or the statement which is wrong is number 3. Move to the next question. Certain protein or messenger RNA that are regionally localized within the unfertilized egg and regular development are called. This is basically associated with the cytoplasmic determinants or the maternal determinants. So the answer will be 3. Next question is, cell-to-cell communication is important in development of an organism. The ability of cell to respond to a specific inductive signal is called. Before answering this, actually the answer is competence, and have some definitions. Here is a definition of induction. Actually, induction is the interaction of the 2 cells which differs in their histories and properties, and the inducer is the tissues that produces a signal that changes the cellular behavior, and obviously the target cell will be the responder, and the ability of cell or tissue to respond to a specific inductive signals is known as competence. So the answer will be competence. Yes, this question. The question is, which of the following does not occur due to stimulation of baroreceptors? Actually, baroreceptors are situated on the inner wall of the arteries, which control the pressure of the blood by controlling the volume or the heartbeat. And the option here, I am little bit confused in those questions, because the first option is bradycardia, which means the decrease in the pulse rate below the normal, and this happens due to the overstimulation of baroreceptor. And second is the hypotension or the low blood pressure. This obviously it would not be there, because baroreceptor controls the blood pressure, by either by controlling the volume of the blood or by vasodilation or vasoconstriction of the vessels. So I think it should be hypotension, but if we will talk about the bradycardia, it is the abnormality of baroreceptors. I'm not very much sure about the answer of these questions, and if you think something or you have some idea about this, then do comment me on my channel in the comment box. I'm waiting for your comment. Please do. Question number 45. Which type of cells located in the gastric cell is responsible for the release of histamine? Actually, G cell is a cell which first releases the gastric enzyme, which then stimulates the ECL or enterochromaffin-like cell, which stimulates histamine, and this histamine will activate the parietal cell to secrete HCl. So obviously the answer will be number 2. Which of the following plant-derived signaling molecules induces hyphal branching or arbuscular mycorrhizal fungi, a phenomenon that is observed at the initial stage of colonization by these fungi? The answer will be number 3, which is trigolactones. Actually, this is the only signaling molecules which are responsible for the hyphae formation in case of fungi. Next question is, vasopressin secretion does not increase with exercise. Now, with exercise and increase intracellular fluid volume, standing and vomiting, vasopressin get increased and secretion increase, and it will affect or target the kidney for the reabsorption of the water from its nephron when the fluid of our body get decreased or the level of the fluid get decreased. And during exercise or any working position or standing and vomiting, our body fluid get decreased, not it get increased. So the vasopressin, when it will not increase in its secretion while the fluid inside our body is more than the requirement. So the answer will be number 2. Next question is, serum has essentially the same composition as plasma except that it lacks. Let's see what the difference is at. Suppose this is a centrifugation tube having blood sample, and if we centrifuge it along with the anticoagulants in it, and when we will centrifuge it, we will get the RBCs and WBCs along with the platelets, or all the cells of the blood get deposited at the bottom of the centrifugation tube, and the transparent materials now we get is the plasma. The serum also is prepared by the same process, only the difference is that in the plasma we are adding the anticoagulants, but serum we doesn't add the anticoagulants. We rather let the blood to coagulate first and then it get centrifuged. So the only difference or the only deficient thing in the serum will be that it will not have the coagulation property. And in the option, Stroth-Trower factor is a type of coagulant factor. So the answer will be number 2. Next question is, which of the following mutagen is most likely to result in a single amino acid change in a gene product? Acridine orange and ethidium bromide. I know that you are also knowing that these two are the dyes which are used for the visualization of DNA strand as it intercalates between the DNA strand, or if these are the base this is DNA strand, then it intercalate in between, and it provide or it will DNA will change the structure of DNA. It will not cause the mutation, and it is irreversible in nature. X-ray doesn't cause the single amino acid change, rather it leads to the dimerization of thymidine. The option will be ethyl methane sulfonate because it leads to it or it targets the guanine, especially on the O6 or the oxygen at the 6th position, and it adds the ethyl group to it. And now the guanine, instead of pairing with cytosine, it will pair with that of the thymidine. Next question is, if non-disjunctions occur in meiosis I, which of the following scenario is likely to occur? The answer will be two gametes will n plus 1 and 2 will be n minus 1. Sorry. Because let's have a look about this. If non-disjunctions occurs during meiosis II, then both of the cells will get affected. Here we will have the 2n plus 2 and 2n minus 2. And so after meiosis II, we will get n plus 2, n plus 1, n plus 1, 2 cell will have the same composition, and the other two will have n minus 1, n minus 1. But when non-disjunction will occur during the meiosis II, as both the cells will have after the meiosis I will might have the normal distribution, but non-disjunction during the meiosis II will lead to the formation of n plus 1 and n minus 1 cell along with the two normal cells, because non-disjunction is not occurring in these cells. So the option will be the first one. So this was in this video. The rest of the questions I will discuss in my upcoming videos, and that will be the end of the explanation of the part B. I'm very thankful for you that you have kept the patience and waiting for my videos, and also I am very much sorry because I delayed so much in explaining whole the question, because I have to prepare the slide and all, whatever the excuses, but I'm sorry. So this much was in this video. Goodbye, have a nice day, and be yourself, and don't forget to subscribe and share this channel. Bye bye.